Good evening to all. Today we are in the second day of this career development program. Pragati madam,
Yes, ma'am.
yes, you can start the program. Ronald sir,
Yes, ma'am.
sir, you are there. We can start the program, sir.
Yes, we can start the program.
Okay. Okay. Pragati madam, you can start the program.
Okay, ma'am. Good evening, everyone. A warm welcome to our respected principal, Dr. R. Sindamarai Madam, Periyar College of Pharmaceutical Sciences, resource person, Dr. A. Ronaldo Anup sir, Silicon Labs Private Limited, today's speaker, Dr. T. Muthukumar sir, faculty members, and my dear participants, for attending 1 week online career development program on computational skills and programming application in pharmaceutical sciences, a hands-on approach. It's my pleasure to welcome you all to this informative session. Programming has become an essential skill in today's digital world, opening doors to innovation, problem solving, and career opportunities across various fields. Now, I invite Dr. A. Ronaldo Anup sir to introduce our speaker for the today's session. Sir?
Thank you, ma'am. Thanks for the opportunity. Dear
Sir?
participants, good evening. Am I audible, ma'am?
Yes,
Sir,
sir.
you are audible.
Yeah, thank you. Good evening, dear participants. It's my distinct honor and privilege to introduce our esteemed first person for today's session, Dr. T. Muthukumar. Is working as assistant professor and associate dean at the School of Sciences and Humanities, SR University, Warangal, Telangana. Dr. Muthukumar has earned his PhD in bioinformatics and computational biology from Vellore Institute of Technology, where his doctoral research was completely focused on identification of potent and selective MAP kinase inhibitors for management of non-small cell lung cancer. His academic journey has equipped him with extensive expertise in bioinformatics, computational biology, molecular docking, molecular dynamic simulation, machine learning, and computational drug discovery. Currently at SR University, he plays a dual role as an educator and an academic administrator. As an professor, he is currently handling courses related to bioinformatics, and as an associate dean in PhD department, he is taking care of the overall administration and management of different PhD programs out there at SR University. Dr. Muthukumar has an established research profile with more than 25 research papers and a cumulative impact factor exceeding 57 and numerous contributions to high impact international journals. His research spans computational drug discovery, transcriptomics, biomarker identification, network pharmacology, and precision medicine. One of the most exciting aspects of his work is his involvement in the rapidly evolving fields of artificial intelligence and machine learning. He is currently working on several AI and ML driven applications in bioinformatics, computational biology, drug discovery, integrating advanced computational approaches with biological data, especially in the case of accelerating the biomarker discovery, identifying novel therapeutic targets, and supporting precision medicine for complex diseases. His contribution has been published in reputed international journals such as Journal of Biomolecular Structure and Dynamics, Pharmaceutics, Molecular Biotechnology, Free Biotech, Molecular Diversity. This reflects his commitment to advancing computational and translational biomedical research. We are truly delighted to have such an accomplished researcher and an academician with us today in this program. I'm confident that his insights and experience will inspire all of us and provide valuable perspectives on the latest advancements in AI, machine learning, bioinformatics, and computational drug discovery. Dear faculty members and students, please join me in extending a warm and heartfelt welcome to our distinguished guest speaker, Dr. T. Muthukumar. Thank you, Dr. Muthukumar. Thanks for accepting our invitation and sharing your valuable time with our participants. The session is yours. Please.
Yeah, thank you, Dr. Ronaldo. So, am I audible, sir?
Yeah, yes, perfect.
Yes, sir. Thank you, sir, for a wonderful introduction. Now I welcome Dr. T. Muthukumar sir, speaker of today's session. The platform is yours, sir.
Thank you, madam. Thank you, Dr. Ronaldo, for the nice introduction. Share my screen. Yes, my screen is visible.
Yes, yes, Dr. Muthukumar.
Yes, sir.
Perfect.
It's
Okay.
fine.
The slide is visible, sir.
Yeah, thank you. I think it is perfectly visible. So I warmly welcome all the participants who are enrolled in this career development program. And I appreciated your effort, even after your working hours, you are joining in this esteemed program to develop your computational skills. So on behalf of our organizing committee, I welcome all the participants for the career development program on the topic of computational skills and programming applications in pharmaceutical sciences. So myself, Dr. Muthukumar, I am working as assistant professor and associate dean. I hope Dr. Ronaldo Anup has given the brief introduction about myself. So we can directly go into the concept. So why the pharmaceutical researchers needed the computational skills? So, and what are the applications we have to utilize for our pharmaceutical applications, and what are the models? Like everywhere we are saying AML, deep learning, so something like that. So what is exactly representing the AI models? So how we can develop AI models, and from where we can develop from the scratch itself. So everything we are going to learn in this upcoming session. So today's session basically a theory oriented session. I hope soon you will get the hands-on training as well. So let me start with a small example. As you have seen, the great example is like polio vaccines. So in early 19, we have suffered with the polio disease. So it took almost 10 to 20 years to develop a standardized vaccine. So today we are getting the polio vaccines for the children. So that disease arrival to vaccine development. So it take almost 10 to 20 years. So at the same time, you can see here like COVID-19. So as the name itself, it is there, COVID-19. So it came in the year of 2019. So within a short span, like within the 1 year of time span, so everyone are able to get the COVID-19 vaccine. So how it happened? So what technology make it happen within the 1 year time frame? So that we have to thank to our pharmaceutical researchers. So and AI as well. So what kind of processing they made to develop the particular vaccines? So without the techniques of computational biology, this would not be happen. So in the upcoming slides, you can see about how we can develop a drug molecules against a particular target protein, and what are the prerequisites it is needed, and how we can test the drug inside the computer. So everything we are going to see one by one, and what are the drawbacks happened in our earlier 19s. So the major drawback is timing. So when the drug has designed in the laboratory, from laboratory to market, how much duration it is taking. So you are from the pharmaceutical background, as you may know that once we tested the drugs in our laboratory, so it has supposed to cross cell line like animal studies. Then we have to go for the clinical trials, phase 2 clinical trials, phase 3 clinical trials. And we have to go for the ethical committee approval like FDA or WHO. So how much duration it taken from the laboratory identification? So in the traditional drug discovery process, it took almost 10 to 15 years of time from the laboratory to market. So there is the major drawback in the traditional drug discovery process. And we are facing a major drawback is nothing but 90% of the therapeutic has drugs as affected with the side effects. So 90% of the drug has a failure rate we have at the earlier stages. So why it is happened? Because when the drug has identified and we cannot test the like admit property like toxicological properties as like today we cannot check it earlier days, so each individual side effects we have to test it in the laboratory and we have to identify those errors like those effects. So but in today's world, with the fraction of second, if we upload the drugs, whenever the laboratory has identified the drugs, if we upload it to some web servers or any AI tool, so it can identify within a fraction of seconds. So what are the possible side effects may happen to that particular drug, or what will happen after 1 year also, whether that particular disease will be recurrence or not. So everything we can predict with the help of our latest technology. So this is the second major drawback. It is nothing but a failure rate. And the third major drawback is like cost. So as we know that many Indian laboratories are lack of funding. So that is one of the major drawbacks what we are facing today. So when you are going for drug discovery in the traditional way, so each individual drug we have to synthesize or we have to purchase it and we have to test for our applications. Like if you want to test the drug against some lung cancer, we have to purchase lung cancer cell lines and we have to test it. So let's say one or two drug, it is possible. If we have like thousands of drug, 10,000s of drug, how we can test all those drugs against the particular disease? So that is not possible, as well as that took a lot of amount to spend it. So in the like in the modern way, we can screen the n number of drugs at a time against a single target protein. So that much technology has developed. So we can test the drug in the computer itself and we can check the activity of the particular drug. Though that also can be done with the help of like tools like molecular docking and molecular dynamics. So if you want to predict the activity, I hope you may heard about the IC50, minimum inhibitory concentration, like how much drug we have to provide to the particular patient and what about the effect of the particular drug. So that also can be done in the computer itself without going for the actual testing. So these three major drawbacks was there in the earlier days. How we are overcoming these drawbacks through the modern drug discovery. So that we will see. So whenever talking about the artificial intelligence, so what it is actually meaning that. So what So in artificial intelligence, we have three different phases of artificial intelligence. The first one is the general artificial intelligence like AI models. So we can call it as AI model. Then we have the machine learning based models like simple algorithms that can read and predict a simple functions. Then we have the deep learning model. Whenever you have the complex data, like two sources of data, like chemical data as well as the genetical data, if you want to combine and analyze for some another applications, let's say for recurrence prediction or drug efficiency prediction. So for that applications, the complex data can be analyzed with the help of deep learning model. For example, CNN, convolution neural network, RNN, GNN, graph neural network, so something on. When it comes to the machine learning model, we have two types of machine learning model. A simple algorithms that can be interpretable with the help of like feature selection approaches. So when we talk about the machine learning models, we have supervised machine learning model and unsupervised machine learning model. So supervised machine learning models are nothing but we have to supervise, like we have to provide the instruction to the machine. You have to perform like this. So let's say if we have the 1,000 number of data, we have to define the 70% of the data is nothing but the training and 30% will be the test set like that. It is like evaluating the models by the own data set itself. So we have to guide the model to perform. So that will comes under the supervised machine learning models. Then the another category is unsupervised machine learning model. So it is mostly working based on the cluster-wise applications. So in unsupervised machine learning model, if we just input the data, that algorithm itself automatically categorize and automatically predict the whatever the our desired output. So that is nothing but unsupervised machine learning model. So in that, apart from this, we have a two major classification, like classification based machine learning models and regression based machine learning models. So what is mean by classification? In the name itself, it is there. Let's say if we want to test the patient has a cancer or not, or which kind of cancer, whether stage 1, stage 2, stage 3, or stage 4, like yes or no, are different classes. So in that situation, we have to go with the classification based models. And then regression based model, if you want to predict the exact values. For example, if you want to predict the hemoglobin level of the particular patient, or if you want to predict the IC50 value of the particular patient, or any kind of like side effect, lipophilicity, or a log, lipophilicity, or a drug likeness. So whatever the things are comes in the numbers. So that can be done in the case of regression type based model. And we have several types of model here also, like graph based neural network. It will work on the molecular graphs, like image based prediction. Then we have a transformer based models. So it is like natural language processing. As you have seen the smiles are nothing but one of the way of representing the drug molecules. So from smiles to how it is converting into the machine readable format, like transformer based model. That model, it will convert automatically the smiles to the numbers. So that is nothing but transformer based model. And we have variational auto encoders. It is like generative models exploring the chemical spaces. So this will help in like drug discovery applications, how the binding sites are located, and based on the binding site, how the drugs are comes and bind to their particular binding site locations. So that kind of interpretation, we can go with the variable auto encoder model. And so for every model, there is an important task from where we can get the data. So whatever the computer applications you are handling, that requires data. So we have an n number of biological databases. As you have seen, almost many databases are available for different applications. So let's say UniProt, like Protein Data Bank, and Ensemble like GenBank, NCPI, and Keck database. So each database is having its own specific function. Let's say if you want to if you want to take the transcriptomic data, it is nothing but like RNA based data. We can go for the NCBI, National Center for Biotechnology Information. There we can get the actual patient data which is already deposited by the researchers around the worldwide. So if you want to study some gene expression or biomarker identification, this kind of studies, we can get the data from the NCBI database. And the second one, the proteomics and structural data. These two are the most important databases. One is PDB, Protein Data Bank, where we can get the three-dimensional structures of the proteins. And when we say the UniProt also the protein database, it contains the raw sequence of the protein. So which is comes under the primary databases classes. So if you want to have the raw protein sequence, so we can get it from the UniProt database. If you want to get the three-dimensional structure of the protein, like 3D structure, if you are going for the binding affinity calculations, so in that cases we can go with the like PDB databases. And we have a chemical data libraries as well. For example, Chembl, then binding DB, and then PubChem also, where we can get the like compounds, like molecules. So the drug molecules. So whatever the drug molecules are approved or investigational or tested in the laboratory. So all kind of chemical associated with their bioactivity. So getting chemical is easy, and getting chemical with the bioactivity data, so that is important thing. So whenever you are going for Chembl or binding DB or PubChem, so many of you know that where we can download the structure of the chemical. But there are hidden informations also available, which is nothing but the bioactivity. So around the world, the researchers are submitting their laboratory results. In earlier days, many people have did like visual screening, like screening quantitative high throughput screening application. So those chemical data along with their bioactivity value are deposited in the Kimball database as well as the binding DB database. If you want to work on the specific disease, let's say cancer. So let's say you are working on the lung cancer. Lung cancer also, we will have so many target protein molecules. So you are selecting any one protein, like you want to target for non-small cell lung cancer and you want to target for the stage 3 or stage 2 lung cancer. So which protein is responsible for the particular type of lung cancer, that we have to identify first. After the identification of the target protein, we have to go and search in this any one of these laboratories. Then we can get the activity data which is done before by the experimental scientists, so that we can get it from these libraries. Then we will have the clinical data. So that is the important thing, is clinical data, from where we can get it, TCGA and Cancer C, and there are many another biostat studies databases. So these databases contain the patient data. So as I said earlier, around the world, researchers are collecting the patient data with the consent of ethical committee approval. They are submitting all these data, like patient data, what is the age of the patient, whether male or female, what is the disease he has come to the hospital, like stage 1, stage 2, or stage 3, whether what is the drug has administered to the particular patient, whether the patient got reoccurrence of the particular disease or not. So all this data will be available in this kind of databases. So how this is useful to develop the models. So with all these databases, we can develop the model to predict some of the biological activity, which is not required to do for the unknown, like newly developed compound, or if you are working with the patient data, you have already developed the model for the prediction of cancer recurrence. So if these are the properties are available, this patient, hello, yeah, if these are the properties of the patient, whether that particular patient will have the cancer again after 3 years, after 5 years. So that also can be predictable with the help of these patient data. We are predicting the future. Basically, we are developing the time machine. So we can say as a machine learning or deep learning or AI model, or we can say it as a time machine. In future, what will happen? That we are predicting with the existing data. So that is one of the major applications with the clinical data. So let me go with the like databases wise. So as I said earlier, Campbell, we have the chemicals around 2.4 millions of compound, like PubChem, then protein data bank, we have the protein 3D structure, then zinc, also some of the databases where you can get the 1.4 million of purchasable compound. Let's say you are screening some applications like, and if you want to purchase the top ranked molecules, that also can be purchased, like from the zinc databases, you will get the cache number, everything, and purchase link as well. And come to the model development. So how the chemicals are representing to the models in the machine learning models. So everybody knows machines can read only the numbers. So in the recent days, we have the natural language processing and graph neural network. So otherwise, machines will read only the numbers. So in graph neural network, you can put the image of the particular data sets. You can directly get the output. And yeah, that is graph representation. And how these chemicals are converted into the numbers. As I said earlier, the smile string. So what is mean by smile string? So let's say salicylic acid, how it can be represented in the form of smiles, or how it can be represented in the form of molecular fingerprints. So both are same chemicals, how we are representing in the different formations. So that is important. Smiles are nothing but like letter-like coding of a chemical, then molecular fingerprints. So the name itself, fingerprints, each chemical having its own fingerprints based on their structural arrangement and its functional as well. So when we talk about the fingerprints, so how it will represent? So basically zeros and ones. So molecular fingerprints are represented by 0 and 1 for all the chemicals. So that is one of the most important thing. And we have the graph representation. Directly we can represent the chemical image, so the system will recognize automatically and can be developed as a model. Then we have the protein embedding, like amino acid sequence can be given as an input for protein embedding, and also we can provide the three-dimensional structure as well for the input for the machine learning models. And we have the domain specific features. It's nothing but if you want to work on like thermostability of the protein, so what are the thermal properties of protein can be included. Let's say if you want to predict the pharmacokinetic or pharmacodynamics or toxicity studies, what are the previous toxicity properties the chemical has, or what are the like basic chemical properties we have. So that also can be given as an input for the machine learning model. So these are all several sample inputs where we can generate with the help of chemicals or protein to given as an input file for the our machine learning model. Let me start with the molecular representations, like little elaborately. We have a two major things. We have one is chemical fingerprint and we have molecular descriptor. So both are different types of representation. When we talk about the fingerprints, as I said earlier, it will be like 0 and 1. Apart from that, we have the molecular descriptors are nothing but the chemical molecular property. Let's say molecular property of the molecular weight of the chemical, how many numbers of hydrogen bond acceptor, how many numbers of hydrogen bond donors, or what is the lipophilicity value, what is the toxicity value, what is the like these kind of properties can be represented in the form of molecular descriptors. So while in the case of chemical fingerprints, it is like binary form 0 and 1. Whether this particular chemical is having the alcohol group or not. If it is present, we can put 1. If it is not present, we can put 0. So something like that. So in molecular descriptors, we have 1D, 2D, and three-dimensional descriptors. That also will be covering in the upcoming slides. So how the chemicals are divided, how these chemical structures can be converted into this 0 and 1s. So initially, we are fragmenting the chemicals. So this benzene ring separately, this alcohol group separately, this particular ring separately. So before starting, we have to fragment the chemical. So this is the form of smiles. So these smiles can be converted in the form of like benzyl alcohol representation. So as I said earlier, how we are fragmenting. It is like circle-based fragmentation. Here alcohol group has fragmented separately, the circle has fragmented separately, and here this particular shape. So this shape also has been fragmented separately. So here we can see benzene group, here alcohol groups, here CH2OH and CH2. So molecular fingerprints will look like this, whether this particular benzene ring is present or not. If it is present, it may be 1. And alcohol group is present or not. If it is present, 1. CH2OH group is present or not. If it is present, 1, like this. So what about this 0? So individual molecular fingerprints it has. So each cell represents individual properties. If it is present, we can mention it as 1. If it is not present, we can mention it as 0. That also can be done. So when we talk about the smiles, how we are representing the chemicals. So initially we will have the atoms. So atoms can be represented like whatever the carbon is carbon like C, O, N, something like that. And we have the bonds. So single bond, double bond, and triple bond. So if the atoms having the single bond, so we have to represent with a small hyphen. So double bond also it is like equal to symbol. Then triple bond we are representing like has kind of symbols. So that I'm skipping a little bit. And types of chemical fingerprints. So we have a 3 types: structural based fingerprints, and property based fingerprints, and then reaction based fingerprints. So what is meant by structural based fingerprints? When we are inputting the three-dimensional structure, the arrangement of structure, so it can be given as a structural based fingerprints. And property based fingerprints are nothing but when we interacting together, like lithophilicity, it is one of the property, hydrogen bond acceptor, how much hydrogen bond donor. So these kind of chemical properties are what is the thermostability of the protein, how much temp like. Oh, so these kind of properties can be comes under the property based fingerprint. Then reaction based fingerprint. From two chemicals are mixing together. So what will be the output? So that is nothing but reaction based fingerprints we have. So when we talk about the structural based fingerprint, these are the example name: Max fingerprint, FCP, ECFP fingerprint, and Morgan fingerprint. Morgan 2, latest we have the Morgan 3 fingerprint, CDK fingerprint. So these are the names of the fingerprint, which has like 2048 rows, as we have seen here. So like this, we have total of 2048 individual structural fragments we have. So that when we apply this Morgan 2 or Morgan 3 fingerprints, so it will be fragmented, a chemical will be fragmented individually, and those all the 2048 properties. So maybe one of the property called oxygen group is present or not, or carboxyl group is present or not, or amine group is present or not. Like that, we have 2048 properties. When we apply to the single chemical, so you can get total of 2048 values, like row values 0 and 1, something like that. So that how we are generating the chemical fingerprint. That's how we are converting a single molecule into the 2048 rows of numbers, whether it is present or not, absent or not. So like that. Then when we comes to the property based fingerprint, as I said earlier, log P is nothing but log P, then molecular weight, then number of hydrogen bond donor and acceptor, and quantum chemical properties also can be considered as a property-based fingerprints. So that also like whether it is present or not. If molecular weight, if somehow molecular weight is above 500 or below 500, if it is above 500, we can represent it as 1. If it is below 500, represented as 0, like that. So whatever the properties, whatever the calculations, if it is fingerprints, so all will comes to 0 or 1. It is like yes or no. Okay. These are the some of the examples, and how we can generate it. What are the tools and softwares where we can give the input, and how we can get that zeros and ones as a like data, like Excel data, we can get it. So one of the main applications or package, we can call it as RD kit. This is like open source software. Whatever the softwares are mentioned here, all are open source based software. There is no need of any license kind of thing. So RD kit is one of the package we require for the conversion of chemicals to the fingerprints. As I said earlier, Morgan fingerprint, CDK fingerprint, and so on. So then we have the open table. It is mostly, you may know that open table is used for conversion of from one chemical structure to another form, like PDB to SDF, PDB to MOL, PDB to MOL2. So open table will use it for conversion. It also has the function to generate the chemical fingerprints. And we have CDK, Chemistry Development Kit. It is an online web server where we can input the molecule, like multiple fingerprints we can generate at a time. So all these software will also integrate with this Java and Python. Mostly the researchers will use Python to integrate it, because further they have to develop the machine learning model with that particular data. Otherwise, it will have like one platform we are generating the fingerprints are, and another platform we are developing the model. So it is like mixed of platforms. It may throw error in upcoming like in further application. So better to use a single source of programming language to calculate or to generate all these fingerprints. So when we talk about the descriptors, so previously what we have seen about the fingerprints like zeros and ones. So here we have the molecular descriptors. So what is meant by molecular descriptor? It has the five different types: zero descriptors, like which don't have any dimers like molecular weight or elemental composition, or how many numbers of atoms present in the particular molecule, or how many numbers of rotatable carbon atoms are available there. Rotatable bonds are available. So that will comes under the 0D descriptors. Then we have the one-dimensional descriptor have number of hydroxyl groups, carboxylic groups, something like that. Then we have 2D descriptors. It is like a small shape kind of structure. And we have the 3D, as we said, three-dimensional structure, volume, surface area, dipole moment, something like that. Then we have the 4D descriptor, where we can measure the dynamic movement of the molecules. That will comes under the four-dimensional descriptor. Here you can see the summary of all the descriptors. Like here we have the drug molecule called aspirin. So I think this may be not interconnected. Okay, so this particular molecule, what is the molecular weight, what is the atom count, and what will be the functional group, like one ester group, one carboxylic group, one aromatic ring. So these are the small values we can get it, and we can represent it as a molecular descriptors. And what are the molecular descriptor tools are available? When we talk about the fingerprint, we have the RDKit or OpenBabel, and we have the CDK, Chemistry Developmental Kit. And that is different. When we want to generate the fingerprint, we have to use all those tools. When we want to identify the descriptors, so here some of the tools are mentioned here. Paddle descriptor, this will work under the use of Java program like modern and dragon and chemoxan also. I think these two we require the license or we can pay also if needed, and DKIT. So these are the tools where we can generate the molecular descriptors. So as per my suggestion, we can go with the paddle descriptor. It also contain 1400 descriptors, different kind of descriptor like 1D, 2D, 3D descriptor it has. It is working on the Java based program. We can install it or we can integrate it with the Python program as well. That maybe you can see in the upcoming this program. And after generating the data, so what we are doing, we are basically developing the model to predict some activity. Let's say if you want to test some drugs against some protein, so how we can identify, how we can predict the activity of the drug without actually testing with the help of AI model. So when we are generating the model, what are the steps we have to follow? So that is the most important thing. Once we have the data partitioning, so that is the important one. So here we have two different types, like one is test data and another one is training data. So training data will helps to train the particular model, and test data will helps to evaluate the model. So whenever we are training something, we have to ask, we have to test the particular model, how much efficiently it is working. That we have to test it. For that, we are separating the test data. And one more kind of data set called it as validation data set. So in training and test data, most of the time it will be comes under the same data set. Let's say if we are having 1,000 data, out of 1,000, 700 will be for training and 300 will be for the test. But all these 1,000 data points will comes under the same sources. So we have to test the unknown source. So that is nothing but the validation, external validation data set. So whenever we are generating the model, our model is supposed to perform in the training, test, as well as in the validation data set as well. So we have to always have three sets of data. Apart out of it, two data set from the same sources and one data set from the different application, like different sources. Let's say we are taking data from Campbell databases. We have to take the validation data from the PubChem database. So that is the kind of data partitioning we will do before starting or before training the model. Then we have the hyperparameter tuning. So each model having its own specific parameters. So why we need to have the hyperparameter tuning? Because whenever you are generating the model, so it will take the standardized like default parameters to give you the predictions. So why that fine tuning is needed? To enhance the model performance or to enhance the prediction. So we cannot easily identify the futures, the characteristics of the model. So that's why this hyperparameter tuning is important. There are several applications called like Optuna hyperparameter tuning or grid search or Bayesian optimization. These are the optimization technique where we can run the model with different combinations of the parameters. So at this end, I will show you some examples how we can perform the hyperparameter tuning based on our research studies. So that tuning algorithm will iterate the model again and again with the mixture of different characteristics of the models. So at the end, we can get the best performing, like best tuned values of the particular model we will get. That is the application of hyperparameter tuning. Then we have the cross validation. So cross validation is nothing but, as I said earlier, we have the 1000 data, 700 will be training and 300 will be for testing. So out of 1000, which 700 will be training and which 300 will be test data set? That we have to that we are initially classifying. So if you mix it, instead of 1 to 700, we can go for 200 to 900. Sorry, 200 to 800. That 200 to 900. So that different combinations of training and data sets, test data sets can be generated, and the models also can be generated. Like usually they will do the 10-fold cross validation. So 10 different combinations of training and test data sets can be taken, and the models will be generated. The average accuracy values can be calculated. So in that situation, so even if your data set is like mixture of data sets, your model can accurately predict the output. That time the cross validation will helpful. And how we can interpret the model? So we are collecting the data and you are generating the data, and how you are explaining your model, how the model itself, yeah, how the model can be explained in terms of feature selection or model features identification. So there are some techniques to identify what are the chemical characteristics which actually helps to predict the biological activity or whatever the desired outcome. So that will comes under the model interpretability. So one of the key term when we are generating the model, the key term is nothing but feature importance calculations, or we have safe feature important calculations. These are the two different approaches where we can identify. Like initially I said we have 2048 fingerprints, like 2048 chemical groups, alcohol groups or hydroxyl group or amide group, out of which all the 2000 group, which group is actually helping for the determining the biological activity. So how we can identify from the model? With the help of feature importance and or shape feature importance calculation. As you can see here, this particular person is highly helping for the prediction, whereas this particular person has the zero contribution in the prediction. Let's see here also you can see this particular person is highly contributing. Let's say this particular persons are very minimal contribution. So like that. So which kind of scaffolds are highly contributing to the particular activity? We are going for a drug prediction, drug activity prediction. So which kind of drugs, which the drug supposed to have the particular scaffold? Without that scaffold, there won't be any activity. Further, you can fine tune that particular scaffold or particular compounds to enhance the further activities. So that also can be done with the help of model interpretability using feature importance and shape values. And model validation, as I said earlier, we have to do the external validation and internal cross validation. So that also can be done. So in a classification based model, we have several parameters to measure the model accuracy. One is accuracy, then precision, recall, F1 score, ROC or AUC, So these are the several metrics used to measure the model performance. When we talk about the regression based models, that can see the R square value or RMSE, mean square error value. So these are the terms or these are the metrics used in regression based models. In classification like accuracy, precision, recall and F1, and regression based model, we have to calculate the R square value. And MCC values are also that can be calculated, Matthew correlation coefficient. It is one of the measure. It can integrate all these performance metrics to calculate the predictive power of the particular model. And how we are developing, after developing the model, how we are developing like websites. So you may see many web servers to predict some of the activity. Let's just admit. So when we are inputting the model, how that algorithm is predicting all the physiochemical properties. So how they have integrated the model into the particular website with the help of API and cloud computing service. They are integrating, and that requires a lot of approval and ethical committee approval as well. And we can even patent also. When you develop a particular model that can produce or that can be efficient in for some specific applications, even you can go for the patent rights as well. And what are the challenges when we are developing the model? What are the challenges we will face it? Bias and generalizability. For example, if you are working on the disease, like whether that particular patient will have disease or not, whether the particular patient having the lung cancer or not, breast cancer or not. Like when we are taking the data, so that time we have to train the model with the equal number of data. Equal number in the sense we have to collect the data who are having lung cancer. Let's say we are taking the 500 patient data who are having the lung cancer. So in another case, we have to make 500 control patients. So this control and disease sample should be equal. When we talk about the IC50 value, whether you are classifying into active or inactive, the equal number of classifications should be there. So that will remove the bias in the data set and that will enhance the model efficiency. And when we talk about the data privacy, most of the clinical data or genomic data restricted to the public concern. Even in TCGA database, we have a two separate category. One category will comes under the public and another one is we have to get the license from the concerned authorities. So data privacy is matter because we are taking the patient data like real-time patient data and we are working on that. So we have to get the consent before we have to work. And some of the regulatory and safety validations are there. And these are the some of our works where we have used machine learning models to discover the drugs. So these two manuscripts which comes under the drug discovery applications. So we have screened over like 12,000 compounds at a time against the non-small cell lung cancer protein targets. And finally we have comes with the like two drug molecules that can be highly efficient against the non-small cell lung cancer. So here also one of the very good article has we have developed a model to predict the lung cancer recurrence. When the patients comes to the hospital for the treatment, so lung cancer treatment, if we are treating either with the chemotherapy or radiotherapy or with some drugs, so what are the probable chance he or she may get after 3 years, after 5 years, whether she will get reoccurrence, whether she will get cancer again or not. So that kind of model we have developed here. And here we have developed peptidase inhibitor prediction. So let us go with a small example how the actual workflow is working. So here initially we have collected the data from the Campbell database. Yeah, Campbell database we have collected over 1084 drug molecules. There 1000 data compounds we have categorized into the active like inactive. Whatever the compounds which are having more than 500 micrometer we have considered as active and inactive we have categorized. After that, as I said earlier, we have a different kind of representation, like different kind of descriptors, different kind of fingerprints. So that we have generated because we don't know which data set, which fingerprint will work. So we have taken all the combinations of fingerprints as well as the molecular descriptors. So that will comes under the molecular representation. So like descriptors and fingerprints. So we have developed 8 machine learning models to predict the activity of the particular drug, whether the drug will be active against a particular protein or the drug will inactive against a particular protein. So here also we may not sure that which particular model has active against a particular protein. So we have developed, so we have generated all the possible fingerprints. We have developed all the possible machine learning models, and we have go with the feature selection. So shape feature selection I have explained already. Then this one RFE, recursive feature elimination. So let's say you have 2000 data points of 2000 molecular fingerprints. Out of how many molecular fingerprints are actually involved in the prediction? Out of 2000, so probably like 300 or 400. What is the exact number of features that will helps to improve the model or that are helps to predict the outcome of the model? So that is nothing but recursive feature elimination is RFE. Then we have Optina for hyperparameter tuning. So to tune the model efficiency. Then we have assessed the model with the help of accuracy, precision, F1 score, and MCC, Matthew correlation coefficient. Then again, we have taken the another data set from the Campbell, like 100 unknown data set, independent data set we have taken, and we have tested with our particular model whether these 100 compounds are actually having active or inactive. So finally, we have compared the actual versus predicted value. So these kind of things we did in the first article. So this article, which is published in the Journal of Biomolecular Structure and Dynamics. Yeah. So, these are the small report of the model. When we talk about the quick properties are nothing but like descriptors, admit descriptors. So with the help of admit descriptors, all the 8 models are generated. Here we have the accuracy value. Like that, fingerprints, we have generated all the 8 models. Then CDK fingerprints again. So out of all the fingerprints and descriptors, here we have the Morgan 2 fingerprints. So which yielded the average accuracy value higher than the other descriptors as well as the fingerprint. So now we have decided, so which fingerprint have the efficiency to predict the activity from this data set. So we got Morgan 2 fingerprints. Still we don't know. So these are the general model with generated with the default parameters. So we have to fine tune it. So which out of which model will provide the efficiency, that we have to fine tune it. For that we have selected the feature selection algorithm. So 283. So as I said earlier, Morgan 2 fingerprint we have 2048. From there, only 283 optimal features are really contributing to the prediction. So that we have identified with the help of recursive feature elimination. This graph you will get it. Then we have the safe feature importance. Here you can see 1856. Let's say the molecular fingerprint 1856 is represented like alcohol group. For example, I am saying, so that alcohol group is highly impacting the prediction of active and inactive. In the case of a chemical activity prediction, like that, we have predicted the top 20 values, which is actually contributing for the active class or inactive class. So here also we have the B swarm plot, which is also represented the each contribution of each bits, like each Morgan bytes, how much it is contributing, whether it is positively contributing or it is negatively contributing, whether it is contributing to predicting the class 0, which is nothing but inactives, or it is helping to predict the class 1 active. So that kind of interpretation we can get it from the help of feature importance calculations. And so hyperparameter tuning also we did with the help of Optina. So here you can see 500 iterations we did. So total of 8 models, sorry, 7 models. So 7 into 5,000. So 35,000 models we have generated, and we have come to the conclusion that this decision tree classifier So these are the exact parameter. Let's say how it will calculate. Let's say max depth is 287. So we are giving some random numbers like 100 to 500. It's supposed to test. So each time whenever the model is generated, so it will set the max depth value, for example, 28 here. Leaf node, it will also have the range of like 500 to 1500, something like that. So each time when the model is generated, all these values will be mixed up, and we can get the final optimized parameters for the particular model. So with that, all optimal parameters, with that like 35,000 models, we have got the seven models with the optimal parameters. And based on the available parameters, we have tested on the test data set. Here we got the light gradient boost model have shown the best accuracy in predicting the biological activities of the non-small cell lung cancer drugs. And this is kind of external validation we did with the external data set, like 100 data sets we have collected externally, and we have tested it again, all the 7 models. So there also you can see light GBM will have shown these 0.7 in the external validation as well. So these kind of predictions can be done with the help of AI models. So how we can generate, how we are generating this model with the help of free source applications. So there are several applications where we can install. For example, like Anaconda. Anaconda is one of the applications where we can install the Jupyter notebook. So Jupyter notebook is one of the application where we can execute our Python code, or where we can develop our machine learning models with the help of our Python code. But every time we have to install the packages. As I said earlier, we have the packages called RDKit, OpenBabel, or like Paddle descriptors. So each and individual packages, we have to install it in our computer manually to perform all these calculations. That is one of the like hardest part, because sometimes the version of your Python version of your computers and the package version it won't match. So that are the challenges we have faced during the time of model generation. So better to go with the latest Python version and Anaconda installer or Jupyter notebook, where you can do it in offline mode. Or if you want to perform, like we don't have that much kind of system, or but still we want to perform the calculation. So the very best idea is nothing but the Google Colab. Google Colab, that requires only your Gmail ID. Easily you can log in and you can create the notebooks, and whatever the codes you want to type, you can run it. And installing the packages in the Google Colab is like a kind of easy task. Like if you put pip install, it will install automatically. So these are all the several packages. I think probably in the tomorrow's session, you will be installing and you will be provided with the hands-on training how how to execute the Python code, or where to execute the Python code. So everything we will be covering in the tomorrow's session. So better we can download any of the packages called Anaconda package or Jupyter notebook, can be downloaded initially before coming to the tomorrow's session. Or if the system configuration does not support this kind of applications, so simply we can have the network and we can log into the Google Colab, and directly we can generate the models. So that's all from my side. If you have any doubts or queries, please welcome. Thank you. I have finished.
Thank you, sir. Pragati ma'am. Participant, the session is open for question and answer. If there is any doubt, you can ask your questions.
Dr. Muthukumar, there's a question in the chat box.
Yes, sir. How
Yeah.
can the novelty of the designed molecules be evaluated? So whenever we are designing the novel molecules, let's say you are like changing the functional groups in the any of the molecules. So instead of synthesizing all the molecules, so we can go with the like molecular docking studies against whatever the applications you want to prefer. Let's say you are designing a novel molecule for some cancer applications. You can select the cancer target protein, and before synthesizing, you can go for the molecular docking or molecular dynamic simulations. So whatever the molecules which are comes as the highest activity, so only that molecules it can synthesize later, as well as you can test the activity of the particular molecules. And what about the reliability of this open source software for detecting IC50 values? So it is not like the open source softwares are nothing but to helps to generate the data. So out of all the descriptors or fingerprints, what I said earlier, so all are reliable. So the reliability questions when it comes. So whenever you are generating the model, so they will test the model reliability, not about the data reliability. Data reliability in the sense from which source you are collecting the initial data and the initial biological activities. So that should be reliable and quantifiable. Like it is not we are going the activity prediction with some 100 or 200 data. So the data set should be larger, and those descriptors or fingerprints you can generate it with the open source software, and model also you can generate with the open source software. But the main task is how the model is working, how efficiently you are fine tuning the model accuracy. So that is important. So most of them are going with the open source softwares only. You have seen the papers which are publishing the machine learning models. Out of 80% are comes in the open source software only. But the only thing is they are introducing some model coupling. So multiple models they can integrate, or the new models also can be generated. So like that. If you use Google Colab, still can be patented, yes, because the coding you are providing. So Google Colab is like a software. So inside, what is the Python code you are providing, or what is the model you are generating? So that is up to you only. That all credits will goes to what whoever is generated. It belongs to them only. This not belongs to the Google. We are just using the platform to generate it, but the idea is ours only. So still it can be patented.
Any more questions, other participants?
Yeah. Thank you, Dr. Muthukumar. Engaging sessions. Yeah, the few questions more. How RAG will help to design the molecules?
I'm not aware of what is RAG. Can you elaborately mention, please?
Sir, can you get more details or can you unmute?
Okay, sir. If you have any more questions, also you can personally text me or mail me so that I can clear your doubts if
exactly.
you have anything.
Exactly. Right. So, of course, thank you, Dr. Muthukumar. This session is like actually we planned like day one, we thought of giving them an information of how the industry is revolving and what are the advancements, what is happening over there. And we have actually tried to predict what is the industrial trend. And day two, we are very happy that you have given a real-time example of how these basics or this Python has actually helped you to come up with some good publications. You have shown, discussed with the participants the output of the programming approaches. And dear participants, from tomorrow, we are going to start with the basics of Python. And during the sessions, we will actually try to begin from the scratch. Before, can we know how many of you are already expertised in any audience who is already expertised in Python programming here in this group? If so, you can share it in the chat box. If you have a prior experience in working, you can please share in the chat box so that we can design the program in such a way. It is actually designed to meet up the students who are really new to this field, who are not aware of anything. Yeah, beginners. Okay, right. So if you could actually share that in the chat box or even in the group, it will be very helpful to us so that we can custom make the presentation based upon the audience setup. As from the information received from the organizers, it's a mixed gathering. We have faculties and students combined over here. Yeah, most of you are beginners, right? So we will do one thing. We will start from the scratch as planned. So from tomorrow, we will be starting it with, as Sir rightly pointed out, we can install all the packages. Tomorrow, we'll give you a hands-on demo of how it has to be done. But most of the sessions we'll be planning in Google Colab, as it is very user-friendly compared to that of installing the packages and everything. We'll give you an information of how both these things can be done. So tomorrow, the session is actually scheduled like understanding the basics of programming, the ABCs of programming. So tomorrow session is actually dedicated to that. And we'll also have an input of all this installation and all the stuff, how to prepare your workstation ready. And from day 4, Monday to Wednesday, we will be dealing with real-time examples, starting from very, very minimal examples of how this programming environment operates. And if possible, we will have a detailed program like how Sarah has mentioned, at least a part of the work which Sarah has done or exhibited to you. We'll try to do a small work on Wednesday. So we request you all to kindly get connected from tomorrow in your laptops or even mobile is, if you are comfortable with, it is okay. Have a practice sessions. We will have a few experiments, means a few programming assignments for you to have practice over that. We'll try to make it as simple as that. So thank you, all dear participants, for being with us and spending your valuable time with us. Thank you, Dr. Muthukumar, for sharing your valuable experience and
Thank you. Thank you.
translating how this can be converted into an excellent research, giving them an idea. Wonderful discussions in the chat box. Meet you all tomorrow again in with the basics or ABC of Python program. Thanks to
Thank
the
you, sir,
entire.
for such an insightful and engaging session. Thank you to everyone for your active participation. I hope today's webinar has been informative and inspiring. On behalf of Periyar College of Pharmaceutical Sciences, I sincerely thank our respected principal madam, resource person, today's speaker, faculty members, and organizer, all the participants for making this webinar a success. Like the sir told, before we wrap up, here's a quick reminder. We will be meeting again tomorrow for the next session. Have a wonderful evening. Thank you. Thank you, sir.
Thank you. Thank you.
Thank you.
Thank you, Dr. Muthukumar. Thanks
Thank you.
to
Thank you, sir.